Hello students, this is your Prasanna ma'am. Today I am here to discuss the topic DNA structure and packaging from the chapter molecular basis of inheritance. After detailed analysis of all the NEET questions and EAPCET questions, we have channelized the topmost important topics for both NEET and EAPCET. From that, the first topic or concept we are going to discuss is DNA structure and packaging. Without much delay, let's get started. Where is DNA present? What is DNA actually? DNA is nothing but the genetic material which is present in all the living organisms. DNA is what? It stores the genetic information of all the organisms. This DNA we will usually call it as polymer of nucleotides, or we can also call it as a chain of polynucleotide chain. Okay, DNA is present both in prokaryotic cell and eukaryotic cell. In prokaryotic cell, what type of DNA is present? In a prokaryotic cell, double stranded circular DNA is present. Mostly it is what ma? Double stranded circular DNA. This double stranded circular DNA in a prokaryotic cell is not bound by any membrane. At the same time, it is not scattered in the complete cytoplasm. This genetic material is located at a particular position in the prokaryotic cell. It is located at a particular site in the prokaryotic cell. Okay, and this is called as nucleoid. How is it present at a particular location without scattering in the entire cytoplasm? It is because the negatively charged DNA is attached with positively charged proteins, which are called as polyamines. Okay, this is the nucleoid which is non-membrane bound or naked, present at one position of the cytoplasm. This is because negatively charged DNA is attached with positively charged proteins, that is polyamines. They held the DNA at a particular position. Apart from this genomic DNA, you have an extra chromosomal DNA, that is plasmid DNA. You have an extra chromosomal DNA, that is plasmid DNA. What is the use of this plasmid DNA in a prokaryotic cell? Plasmid DNA plays a very peculiar role. For example, this gives a specific phenotypic feature, phenotypic character to the bacteria, specific phenotypic feature to the bacteria. For example, resistance to antibiotics, and it also plays an important role in sexual recombination. Okay. Next question. In eukaryotes, DNA is in the form of double-stranded linear DNA. DNA is in what form? Double-stranded linear DNA in eukaryotes. In this eukaryotic cell, complex compartmentalization of each and every organelle is seen, wherein DNA is present in double membrane bound nucleus. That DNA is highly condensed in the form of chromosome. This complete condensation of DNA into chromosome takes place in which phase of the cell division? It takes place in metaphase of cell division, wherein DNA inside this chromosome is in what form? Double helical, double stranded linear DNA. Double helical, double stranded linear DNA you will find in eukaryotes. It is highly condensed in the form of chromosome. This is about DNA. Now we have learnt that DNA is a polynucleotide, right? What is nucleotide actually composed of? Nucleotide is composed of sugar, nitrogen base and phosphate group. Nucleotide is composed of what beta? Sugar, nitrogen base and phosphate group. What type of sugar? What are different types of nitrogen bases? How these are binded to form a nucleotide, and how this forms a polynucleotide, single strand and double stranded polynucleotide chain we are going to see in the coming slides. Okay. Before that, look into the length of DNA present in different organisms. How do you identify the length of DNA? What is DNA? Which is nothing but polynucleotide chain, no? If a single nucleotide chain is present, if a single nucleotide chain is present, it is called as single stranded DNA. Okay. If two nucleotide chains are present, two nucleotide chains are present which are held by hydrogen bond between bases. Okay. What are they? We are going to see now. Okay. Double stranded DNA. This is what known as double stranded DNA. How we will identify the length of DNA? Length of DNA we can find either by number of nucleotides if it is single stranded DNA, or number of nucleotide pairs, number of nucleotide pairs or base pairs, nucleotide pairs or base pairs if it is double stranded DNA. Just by looking into the nucleotides or pairs we can identify whether it is single stranded or double stranded. If you look into the bacteriophage, pi into 174, it has how many nucleotides ma? 5386 nucleotides, means it is single stranded or double stranded? It is single stranded circular DNA. Okay. If you look into bacteriophage lambda, it has base pairs, means it is double stranded circular DNA. It is what ma? Double stranded circular DNA. If you look into E. coli, 4.6 into 10 to the power of 6 base pairs again. Here it is again double stranded circular DNA. Okay. If you look into haploid content of human DNA, that is 3.3 into 10 to the power of 9 base pairs. Here it is eukaryotic cell, right? So it is double stranded linear DNA. Okay. So how nucleotide, each nucleotide has what ma? Sugar, nitrogen base and phosphate group. They are connected as such and forms a single stranded DNA. This is what? Single stranded DNA, where it is counted in the form of length of DNA is calculated by number of nucleotides. Here as it is in two stranded, it is calculated in what form beta? Number of base pairs or number of nucleotide pairs. Next, if you see, it is nitrogen bases. Nucleotide is composed of sugar, phosphate and nitrogen base, right? Now first we will learn what are the types of nitrogen bases present. What are the types of nitrogen bases present? Two types beta. One is pyrimidines, and the second is purines. What are them? Purines. If you look over here in the pyrimidines, it is single cycle. Okay, pyrimidine is what? Single cycle, and purine you will see two cycles attached. Okay, pyrimidine is one cycle and purine is two cycles. And the number of nitrogens present in a pyrimidine are at first site and third site. First and third site. Okay, at 1 and 3 positions nitrogen group is present. If you look over here at 1, 3 and 7, then 9. At 4 sites or 4 positions nitrogen base is present. Okay, at 4 sites or 4 positions what is present? Nitrogen base is present. Okay, what are different types of pyrimidines we have? Pyrimidines that occur in DNA. Pyrimidines that occur in DNA are cytosine and thymine. Pyrimidines that occur in RNA are cytosine and uracil. Thymine doesn't occur in RNA. Here what are they? Cytosine and uracil. In DNA it is cytosine and thymine. Thymine has a synonym which is given in our book, that is 5-methyl uracil. What is that beta? 5-methyl uracil. 5-methyl uracil. Okay. Next, purines both in DNA and RNA. Both in DNA and RNA. What are the types of purines we see? Those are adenine and guanine. Okay. These are present both in DNA and RNA. Next, if we see the clear distinction or differentiation between purine and pyrimidine, just now we have discussed. You can take a screenshot if you want and recall it properly. Next, types of sugars both in DNA and RNA. What type of sugar we will see? We have two types of pentose sugars beta. We have two types of pentose sugar. Pentose sugar means 5 carbon, right? Pentose sugar means how many carbon? 5 carbon. As such, we have two types of sugars. One is ribose and the other is deoxyribose. If you look into ribose sugar, I said you how many carbons? 5 carbons, wherein C1, C2, C3, C4 and C5. Here also 5 carbons, C1, C2, C3, 4 and 5. If you look over here, at third carbon the hydroxyl group is present in both the sugars. At third carbon hydroxyl group is present in both the sugars. This forms 3' hydroxyl end in a double stranded DNA. 3' and 5' is present now. At 3' hydroxyl group is this only in both DNA and RNA. At second carbon oxygen group is absent in deoxyribose sugar. That is why the name is what ma? Deoxyribose. Here oxygen group is present, that is ribose sugar. Next, if you see nucleoside formation, how a nucleoside is formed? Nucleoside is formed by nucleoside is nothing but attachment of nitrogen base with sugar. Okay, with sugar. Okay, C1 of pentose sugar is attached to N9 of purine. C1 of pentose sugar is attached to N9 of purine. Forms a, how is it attached? It is attached through glycosidic bond. How is it attached ma? It is attached by a glycosidic bond. We will call it as what? N-glycosidic bond. N-glycosidic bond. And in a pyrimidine, C1 of pentose sugar is attached to N1 of pyrimidine. C1 of pentose sugar attached to N1 of pyrimidine. As such, what type of nucleosides you will see in DNA and what type of nucleosides you will see in RNA. In a DNA you will see deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine. These are the 4 types of nucleosides you will see in a DNA. What are the types of nucleosides that you see in RNA? Adenosine, guanosine, cytidine and uridine. And what beta? Uridine. When nitrogen base, that is uracil, attached to ribose sugar, it forms what? Uridine. Next, we have understood what is glycosidic bond. Next, here we need to understand 2 more important bonds for the formation of nucleotide and polynucleotide. One is phosphoester bond. What is that ma? Phosphoester bond. Phosphoester bond. Phosphoester bond is formed between C5 carbon and phosphate group. C5 carbon of pentose sugar and phosphate group. This is nucleoside. What is this? This is nucleoside. Sorry. This is nucleoside. sugar and nitrogen base. Sugar and nitrogen base forms a nucleoside. Nucleoside's fifth carbon is attached to phosphate group forming an ester bond. You will call it as what beta? Phosphoester bond. This nucleotide is attached to the another nucleotide when 3' hydroxyl group is attached to the phosphate group. Here what is attaching? 3' hydroxyl group is attached to phosphate group. These two ester bonds together forms what beta? Phosphodiester bond, which forms a dinucleotide. Which forms what? Dinucleotide. Okay. Now we have clearly understood the types of bonds which are involved in the formation of nucleoside, nucleotide and a dinucleotide. Okay. Next. Yes. If you look into, if we recall what are the types of bonds? Bonds involved in nucleotide in polynucleotide chain formation. Polynucleotide chain formation. Okay. First, the bond between sugar, that is pentose sugar and nitrogen base. Okay. If it is sugar, that is either ribose or deoxyribose attaching with purine, it is C1 and N9, right? It is C1 and N9 glycosidic bond. If it is with pyrimidine, C1 and N1 glycosidic bond. So, nucleoside includes glycosidic bond. What bond ma? Glycosidic bond. Second type of bond, that is phosphoester bond. What is phosphoester bond? Nucleoside. From nucleoside, fifth carbon of sugar is attached to phosphate group. Is attached to what? Phosphate group. This forms phosphoester bond. What does it form? Phosphoester bond. Okay. Two nucleotides are attached to each other by phosphodiester bond. What is that bond? Phosphodiester bond, wherein one ester bond is in between C5 carbon and phosphate group and the other ester bond is in between 3' hydroxyl and phosphate group of the other nucleotide. This is one nucleotide attached to another nucleotide by these two ester bonds. That is why this is called as what ma? Phosphodiester bond. So, if the question is what are the bonds involved in nucleoside, glycosidic bond, what are the bonds involved in the formation of nucleotide? Nucleotide formation includes two bonds, one is glycosidic bond and second is phosphoester bond. Third question, what are the bonds involved in the formation of polynucleotide chain or dinucleotide? Then three bonds you need to include, that is glycosidic bond, phosphoester bond and phosphodiester bond. If in case the question is like how many bonds are included in formation of double stranded nucleotide chain, double stranded polynucleotide chain. In that case, one more bond is added, that is hydrogen bond. What is that ma? Hydrogen bond, wherein two bases of two stranded nucleotides are connected inside. Okay. For a double stranded DNA, four bonds are required. What are they? Glycosidic bond, phosphoester bond, phosphodiester bond and hydrogen bond. Yes. Next, if we look into Watson and Crick model of double helix model, we will call it as. Okay, firstly if we look into the discovery, in 1869 it is Frederick Mister. In 1869 Frederick Mister discovered DNA as an acidic substance present in the nucleus as nuclein, and he named it as what ma? Nuclein. Later X-ray diffraction studies were conducted by Rosalind Franklin and Maurice Wilkins. From these X-ray diffraction studies, Watson and Crick in the year 1953 proposed a model called as double helical model of DNA. What is this double helical model of DNA? What does this model say actually? He says that a particular double stranded DNA has total number of purines, that is adenine and guanine are equal to total number of pyrimidines. Total number of purines are equal to total number of pyrimidines. Okay, this is the first point you need to remember, which is derived from the double helical model given by Watson and Crick. Okay, most important. Second point, what did they say? The number of A plus T by G plus C. Adenine thymine by guanine cytosine ratio. The ratio between adenine thymine, guanine and cytosine is specific for each species. This is specific for each species. Specific and constant for each species. If you take a prokaryotic cell or prokaryotes, the base constant or the ratio of this is less than 1. It is what ma? Less than 1. It is less than 1. For example, in an E. coli, it is around 0.92. The base ratio is what? Around 0.92. For example, in a human cell, it is around 1.52. Okay. Next. Here if you see the features of double helix of DNA from the points which Watson and Crick has given, that a specific ratio between a specific base ratio of A plus T by G plus C, and a DNA include equal amount of purines and pyrimidines. Number of purines are equal to number of pyrimidines, and in a polynucleotide chain the number of sugars are equal to number of phosphate groups. The number of sugars are equal to number of phosphate groups. These are the points given by double helical model of Watson and Crick. From that, salient features of DNA were driven. What are they? It is saying like two polynucleotide chains where the backbone is constituted by sugar and phosphate. If you take a double helical structure of DNA, at the center we have already discussed, the nitrogen bases are present. What are present? Nitrogen bases. For example, adenine thymine, guanine cytosine, again adenine thymine, CG, TA, AT, like that. Okay. So, purine is connected with pyrimidine. At the center, two nitrogen bases. If it is adenine, it has two hydrogen bonds between thymine. Adenine and thymine are bounded by how many hydrogen bonds? Two hydrogen bonds. Guanine and cytosine are connected by 3 hydrogen bonds. These hydrogen bonds and nitrogen bases are at the center, wherein the sugar and phosphate group are the backbone of DNA. Sugar and phosphate group form what beta? Backbone of DNA. Next, the two chains are coiled in a right-handed fashion. Two chains are coiled in what fashion? Right-handed fashion, that is B type of DNA. The most stable DNA, which is B type of DNA, it is coiled in right-handed fashion. The pitch of DNA is 3.4 nanometers. What is pitch of DNA? One complete turn of DNA. one complete turn of DNA from here to here, it is called as pitch of DNA. That is 3.4 nanometers or 34 angstroms. What is that? 3.4 nanometers or 34 angstroms. And distance between two base pairs, is 0.34 nanometers. Distance between two base pairs is how much ma? 0.34 nanometers. The plane of one base pair stacks over the other in the double helix. The hydrogen bond we have discussed, right? A connects with T of the other strand by 2 hydrogen bonds, G with C by 3 hydrogen bonds. As such, one base is stacking over the other base, and this maintains equal distance between two strands of DNA. Next. Yeah, if we see as adenine pairs with thymine, guanine with cytosine, this maintains the equal length or equal size of the polynucleotide chain, wherein these two chains are anti-parallel to each other. These two chains are what? Anti-parallel to each other and complementary to each other. They are anti-parallel and complementary to each other. This is called as anti-parallel polarity, and they are maintaining complementarity also. The hallmark of Watson and Crick's double helical model is nothing but base pairing. It is what ma? Base pairing. This is the hallmark of their proposition. Based on this, they have derived many points that we have just discussed. Okay, the bases in two strands are paired through hydrogen bonds. Yes, that forms the base pair. Okay, these are connected by hydrogen bonds, and this forms the base pair. This forms what ma? Base pair. Next if we see, after knowing the double helical model of DNA given by Watson and Crick and salient features of DNA, Watson and Crick has proposed the central dogma of molecular biology. Central dogma of molecular biology. Okay, what is the central dogma of molecular biology? The DNA which is existing in a particular cell first replicates. Okay, DNA replication or duplication happens in a semi-conservative way, wherein DNA duplicates, and then the information which is in DNA is transcribed to RNA. Information which is in DNA is transcribed to what beta? RNA. From that, messenger RNA, rRNA and tRNA combinedly synthesizes proteins. The information is translated to proteins. So, first the ultimate goal of DNA is what? Synthesis of proteins. DNA to RNA, RNA to proteins. That is the central dogma of molecular biology. But there are few exceptions. For example, in retroviruses, there are few viruses. What are they? Retroviruses like HIV, where the genetic material is RNA. RNA cannot directly give the protein, right? So, this RNA have to convert into protein by reverse transcriptase. By what ma? Reverse transcriptase enzyme. Reverse transcriptase. This is called as reverse transcription. After DNA is obtained, from that the protein is delivered ultimately. Okay. Next, we will learn the packaging of DNA. Packaging of DNA. If we look into DNA packaging, what is the necessity of DNA packaging actually? DNA, if you look into the length of DNA, I already told you. How can you calculate the length of DNA? Length of DNA can be calculated. Length of DNA can be calculated by number of base pairs into distance between two base pairs. Number of base pairs into distance between 2 base pairs. If you consider a human cell, number of base pairs in a diploid human cell is 6.6 into 10 to the power of 9 base pairs, wherein distance between 2 base pairs we have just learned, that is 0.34 nanometers. If you convert it into meters, 0.34 into 10 to the power of minus 9 meters, 10 to the power of minus 9 meters. Just a second. Yes, 0.34 nanometers, that is 0.34 into 10 to the power of minus 9 meters. Together, if you look into, the complete length of human DNA is 2.2 meters. Okay, this is how you will calculate the length of DNA. If for suppose number of base pairs are given and he is asking us to find out the length of DNA, then what will be the formula? Number of base pairs is equal to length of DNA by distance between 2 base pairs, by distance between 2 base pairs. This is very important formula, please do remember. We will later come into the calculation part also. So, if you see packaging of DNA, why is packaging necessary for a DNA? If you look into the length of DNA, which is around 2.2 meters long, that is 6 feet long, it has to fit in that small nucleus. For example, in a human cell, the nucleus is of 10 to 20 micrometers in diameter. So, to fit into that small nucleus, DNA has to be packed properly. How does DNA package happen in both prokaryotic and eukaryotic cells? Both prokaryotic and eukaryotic cell. Prokaryotic and eukaryotic cell. In a prokaryotic cell, if you see, in the beginning we have discussed, right, DNA is snaked at one position in the nucleoid form, where negatively charged DNA are attached by positively charged polyamides, and this coils or condenses the DNA at one position and you are calling it as nucleoid. This is the only simple DNA packaging which happens in the prokaryotic cell, wherein negatively charged DNA is attached with positively charged polyamides or the positively charged proteins. This packs it into nucleoid in the cytoplasm of a prokaryotic cell. But in a eukaryotic cell, the DNA packaging has for DNA packaging many models are given, but the most accepted model is nucleosome model, wherein negatively charged DNA is combined with positively charged histone proteins. What proteins ma? Histone proteins, and this condenses it to chromosome. The levels of packaging include as such in a eukaryotic cell. First is supercoiled DNA, then chromatin form, which you will see in the prophase of cell division, then DNA along with histones. Complete condensation happens in the metaphase. Complete condensation happens in what phase? Metaphase of cell division. This finally gives the DNA double helix structure. These are the levels of packaging in the eukaryotic cell. If you detailedly see what are the histone proteins, how are they packed? Here DNA is negatively charged, we already know that. It is coiled around positively charged histone octamer. Why is it called as histone octamer? Why is histone basic? Because histone is filled with basic proteins like arginine and lysine constitutes in the histone. Histone constitute what basic proteins? Arginine and lysine amino acids. Okay. Now, histone has 4 type of histone proteins. What are they? H2A, H2B, H3 and H4. 2 sets of this. How many sets beta? 2 sets of this will form histone octamer. To this histone octamer, the negatively charged DNA is coiled or wired in this form. In this form it is coiled, okay. Okay, in this form it is coiled, and to one histone octamer and the other histone octamer, linker DNA is present. What is present? Linker DNA. Linker DNA. This linker DNA is of H1 histone protein. It is of what protein? H1 histone protein. This combined nucleosome contains how many base pairs of DNA? It contains 200 base pairs of DNA. Okay, it contains how much base pairs? 200 base pairs of DNA. Next, further condensation into chromosomes, further tight condensation into chromosomes involves some special proteins called as non-histone proteins. What are they? Non-histone proteins. They play a very important role in organization and compaction of chromatin into chromosome. Okay, for chromatin, this histone proteins and DNA is wrapping like this. That gives you an appearance called as beads on string appearance. Beads on string appearance. What are those beads? Beads are nothing but histone octamers. On that it is giving beads on string appearance. That is seen in chromatin. In what ma? Chromatin. Chromatin. Further condensation into chromosomes is done by some non-histone proteins. It is done by what beta? Non-histone proteins, which further condenses it into chromosome. Which further condenses it into chromosome. Okay. Next, after chromosome formation, you will see 2 types of chromosomal packing. One is euchromatin. One is what ma? Euchromatin, and the other is heterochromatin. One is euchromatin and the other is heterochromatin. In euchromatin, the DNA is loosely packed. It is loosely packed. And here DNA is easily accessed by RNA and proteins. It is easily accessed by RNA and proteins, and it transcription and translation easily happens. Transcription and translation occurs. Translation occurs. As it, and this is loosely stained. It is what beta? Loosely stained or lightly stained. It is lightly stained. Okay, these are the 3 important points you have to remember with respect to euchromatin. Heterochromatin, if you consider, this is tightly packed. What is it? This is tightly packed. Tightly packed, and RNA and proteins cannot access the information present in this chromatin region. RNA and proteins cannot access the information present in the DNA, and this is darkly stained. This is darkly stained. These are 2 types of chromatin which are formed at the end of packaging. One is expressed and the other is unexpressed and dark. Okay, darkly stained. This is what we have learnt today, that is DNA structure and packaging. Tomorrow we will be coming up with most important topic again from the molecular basis of inheritance. Thank you.